Prior to 1958, there was a lot of speculation about how DNA replicated. There were three proposed models of DNA replication. One was the conservative model of replication, two was the semi-conservative model of replication, and thirdly was the dispersive model of replication. For the first one, the conservative model of replication, this suggested that the two strands that made up the parental helixes would somehow join back together and form one daughter cell, which was consisting of both of those strands in the parental helixes, and another strand, another daughter helix, which consisted of newly synthesized DNA. All right. And for the dispersive replication model, this suggested that the parental helix, or DNA helix, would somehow break up into different segments, and each individual segment would act as a template to make the two daughter DNA helixes. Thirdly, we have the semi-conservative model of replication. In this model, it was suggested that the parental DNA helix would split up into two individual strands, and each strand will act as a template to form the newly synthesized DNA. And thus the two daughter helices will consist of one strand of DNA from the parental helix and one newly synthesized strand of DNA. In 1958, Matthew Meselson and Franklin Stahl designed and performed an experiment which proved the semi-conservative model to be correct.
I'm Stahl.
I'm Meselson.
And together, we are Meselson and Stahl. So today, we're going to prove that semi-conservative DNA replication is the true form of DNA replication. And to do so, we have E. coli in an N15 gradient that we will later compare to an N14 gradient. So what we're going to do first to separate the N15 DNA out of the E. coli is we're going to have to break it down in a centrifuge. We have the N15 DNA, which we've isolated through centrifugation. And what we're going to do is we're going to put it into N14 medium, which is naturally occurring. The cells have grown, and now we're going to extract DNA.
We have for generation 0 a pure N15 DNA, and this is what it looks like.
For generation 1, what's going to happen is the N15 DNA is going to split, and because while previously this DNA was in a purely N15 medium, it's now in a purely N14 medium. So that means instead of making N15 DNA, it will make N14 DNA. And this process right here disproves conservative DNA replication, because essentially what conservative says is that we'll have two old DNA molecules or strands and then two new ones will form. But during this first stage, we can see that it actually pairs up and we have two hybrid strands. Alright, so generation 2 of our centrifugation, we start with two hybrid strands of DNA, and during centrifugation that splits, and because once again we're in a purely N14 medium, it is replaced by N14 strands, which this method proves semi-conservative DNA replication because there's a 3:1 ratio of N14 to N15 DNA strands.
This little number right here disproves the dispersive theory, because in the dispersive theory there aren't any pure strands of N14 DNA.
In 1953, Watson and Crick came up with a semi-conservative replication hypothesis.
They couldn't prove that the semi-conservative replication hypothesis was right, but in 1958, Meselson and Stahl's experiment proved that their theory was right, which is why it's important.